Welcome back to the course Fundamentals of General Immunology, and we discussed in the last class about the transcription control of B cell, where we looked into different transcription factors, how these transcription factor expression and migration towards the nucleus can modulate multiple genes transcription, which can lead to production of different proteins, which can eventually do functionality in terms of differentiation, more production of the proteins such as antibodies, and also de-differentiation and memory immune response. So this is typically a control mechanism in terms of gene expression, how these genes are transcribed to produce mRNA, effective mRNA, to get the function. So in the same way, there are transcription controls that's happening in the T cell population as well. So we'll try to understand this transcription control in the T cell population in today's lecture. Before starting, we need to recapitulate that there are multiple subsets of the T cell, such as T helper cell population. In T helper cell also we have Th1, Th2 and Th17. Apart from that, we have cytotoxic T cell population and also T regulatory cell population, which involves in suppression of the immune response, particularly by expressing TGF-beta and IL-10 like cytokines. So we'll try to look into details of the all different cell types signaling transduction in terms of transcription control. So let's start the process of the T cell development. So right from the thymus, so we know that thymus is the site for T cell production and the bone marrow is the site of B cell production. So if we consider thymus, in the thymus we always see that there could be a progenitor cell which we call hematopoietic. Hematopoietic stands for the cells which are going to form the blood cells, particularly the immune cells. And after their differentiation, these hematopoietic progenitor cells are converted into double negative. We call it DN, where the CD4 and CD8 both are negative. And this is just a process of development. And then we can see that there are multiple transcription factors which are regulated in order to make the cells as double negative. And then there could be a lineage commitment. And then after the positive selection, we can get double negative to double positive selection, where we can get CD4 plus and CD8 plus T cell population in the thymic level. And after that, we will see that there could be a further round of selection where we can see either CD4 plus or CD8 plus single positive cells are selected, and these selected cells are ultimately going to give the immune response in general. Now, these are the way T cells are getting generated in the thymus, particularly in terms of the expression of the protein. So when we say the expression of the protein such as CD4 and CD8, that stands for these are critically regulated. So suppose if there is double negative, that stands for there is no production of CD4 and CD8, and then suddenly there is an expression of CD4 and CD8, and then again there could be a single positive cell where there could be a suppression of CD8 plus expression, CD8 expression in a particular cell, or CD4 plus expression in a particular cell, in order to make the cell as a single positive. That means these all protein regulation and protein production regulated by the level of mRNA from the nucleus, and which is what is transcription control. So let's try to see what are the things which are responsible for it. And the key transcription factors which we have discussed earlier also that are responsible for thymic selection is Notch1. And this Notch1 we have discussed in great detail. So this can inhibit the B cell fate from a cell type, and also it can induce T lineage commitment. So this Notch1 pathway is important transcription factor. There are other proteins which also play a role, such as E proteins. E proteins is actually E2A, HEB. These are actually basic helix, loop helix kind of transcription factor. These are also called as E proteins, and this acts as a crucial regulator of lymphocyte development and differentiation. So this can promote also T cell lineage and control proliferation and allelic exclusion. So these are proteins which are actually involved in regulating the gene expression. And finally, we have Icaros, which is IKROS, which is coming from Icaros family zinc finger protein. So these are actually a hematopoietic specific transcription factor, which are specifically expressed in the cell which are going to form the B cell and the T cell population. So these are actually a very specific kind of transcription factor which are regulated at the hematopoietic level. So these are essential for early development of T cell, and also it regulates the chromatin accessibility and lineages gene. So that means it can cause a lot of changes in the chromatin level, because when we say chromatin, that means the DNA is tightly wrapped around with the structure called histone protein. So there are complex of histone protein which we call solenoids, and these histone proteins, since these are the proteins are basic charged, and based on the charge interaction, since the DNA is negatively charged, these are tightly wrapping the DNA around it. So basic protein histone is having, so suppose this is a basic protein, so DNA is coiled around this structure. So this is histone and this coiled structure is DNA. And these structures are actually tightly regulated. So in order to make transcription happen, the DNA has to unwind. And because of that reason, we always see that there could be a lot of modification that's happened in case of histone protein in order to make histone detached from the DNA in order to do some transcription. And therefore, we always see that chromatin remodeling, that is what we call, some of the chromatin remodeling stands for epigenetic controls, are also part of it. So this is very important protein called Icaros, which basically involved in hematopoietic specific transcription regulation, and of course these can help in lymphocytic development as well. So this is basically the level of control that is happening in case of thymus. So the hematopoietic progenitor cells are going to form into CD4 plus or CD8, never both, in a very synchronous way by this transcription factors such as Notch1, E proteins, and Icaros. So we'll see how this peripheral level, actually the T cell is getting selected or transcriptionally controlled. So when we say peripheral level, we looked into just now in thymic level, which is a central level. So thymic is basically the organ where the things are formed, and then post development these T cell which are CD4 plus or CD8 plus are going to the periphery to the function. Now again going to the periphery, so suppose this is a thymus organ, and from there the T cell which are lineages CD4 plus, or there could be CD8 plus. Now these cells are going towards the periphery, and in periphery they are also doing certain functions. Now to do that function, they need to again do a lot of transcription control. So let's try to understand what is happening for the 4 plus cell and 8 plus cell. So we'll see it separately. Let's try to focus on the 4 plus cell, which is also called as helper T cell. Now these are the level of helper T cell regulation in the periphery. So once there is antigen co-stimulation, so antigen presenting cells are coming in the site, and once they are sensitizing the naive CD4 plus T cell, these are differentiated into either Th1, Th2 or Th17. Apart from that, we also looked into two other cell types which we'll discuss here. So we looked into CD8 plus cell, which is also called as cytotoxic T cell, and we'll also look into another cell type which is called as T regulatory cell, which are actually positive in FOXP3 transcription regulation. So these are the proteins which are specifically regulated in the T regulatory cell. So these are actually the markers. So let's write here. So this will be FOXP3. So these are the three proteins which can help us to delineate that which are the different cell type. So once we are talking about T helper cell here, so these are differentiated into either Th1, Th2 or Th17. So let's focus one by one into individual subtypes. So let's first look into Th1. So in Th1 differentiation, the Th1 differentiation is depend upon the expression of or the secretion of very specifically IL-12 and interferon gamma. So these are the two cytokines which are secreted during the Th1 differentiation, and post expression of these IL-12 and interferon gamma, there is an expression of T-bit. T-bit is actually a T box expressed in T cell transcription factor. So we could see the T-bit in a very unique way that these are Th1 specific transcription factors. So once T-bit is expressed as a transcription factor in the cells which are differentiated with IL-12 and interferon gamma, then we can see that there could be an expression of interferon gamma, TNF, gene TBX21 and CXCR3, which is a chemokine receptor, particularly in the Th cell population. So these are actually differentiating the Th1 cell type, which is responsible for intracellular pathogen killing, so such as viruses or some of the intracellular parasites such as Mycobacteria tuberculosis or Listeria monocytogena. So we will discuss when we will talk about the pathogen which are intracellular in nature. Intracellular stands for inside the cell. So all the viruses, most of the viruses are actually intracellular in nature, while few bacteria such as Mycobacteria tuberculosis and Listeria monocytogena are actually intracellular. So Th1 does play a very important role in clearing those pathogens. So remember, T-bit is very important transcription factor for Th1 differentiation. Now let's focus on Th2 differentiation. In Th2 differentiation, the major cytokine which decide the Th2 proliferation is IL-4. So each differentiation is distinct from their type based on the cytokine. And the transcription factor which comes into picture is GATA3. GATA3 stands for GATA binding protein 3, which is again a very critical transcription factor that cause expression of IL-4, IL-5, IL-13 and CCR4, again chemokine receptor, very specifically CCR4. Now these are very typical cell type which does protect the immune host body from helminthic infection, parasitic infection and allergic infection. So these cytokines IL-4, IL-5 and IL-13 is very unique to helminthic infection. Or eosinophilic based infection where allergic reactions are coming into picture. So Th2 differentiation is based on IL-4 expression, GATA3 transcription factor and production of these cytokines such as IL-4, IL-15, IL-5 and IL-13. So this is exactly the way we will see in Th17 as well. So Th17 is getting differentiated because of the expression of transforming growth factor beta, IL-6 and IL-23. So these cytokines are different as compared to Th2 and Th1. So TGF beta, IL-6 and IL-23 causes an expression of ROR gamma T, which stands for retinoid related orphan receptor gamma T, which is very unique transcription factor in Th17 differentiation. And the differentiated Th17 cell will probably in long term will express IL-17A, IL-17F, IL-22 and CCR6 cytokines. So IL-17 and 22 is very unique to Th17 type, and this Th17 is actually responsible for immunity against extracellular bacteria and fungi. So we'll see again in detail about their role eventually when we'll discuss about the fungi or extracellular bacterial infection, that how this Th17 also it regulates the gut homeostasis to a great extent, which we'll see later classes, that how this Th17 expression of IL-17 or 22 can modulate those effective immune response. So let's summarize the whole process. So we are trying to look into the cells which are differentiated from T helper cell. So there are three types which we are discussing here: T helper 1, T helper 2 and T helper 17 cells. These are controlled by different cytokines, and for Th1 it is IL-12 and interferon gamma, for Th2 it is IL-4, and for Th17 it is TGF-beta, IL-6 and IL-23. And in turn they are producing three different transcription factors in the cell type. Th1 can produce T-bit, Th2 produce GETA3, and Th17 produces ROR gamma T, which could lead to differentiation of Th1, Th2, Th17 cell types to do different function. Th1 can only involve or mostly involve in intracellular pathogen clearance, while Th2 is mostly on helminthic infection and allergic reaction, while Th17 is mostly towards the extracellular bacteria and fungi, and they are tightly secreting few important cytokines which are listed here. So 17 and 22 is for Th17, IL-4 and 5 particularly for Th2, and interferon gamma is very specific for Th1 cell. So these are some of the things which are differentiating the T cell in the periphery, particularly T helper cell. Now let's try to see what is happening to the cytotoxic T cell population. Again, there will be a different set of transcription factor that could regulate the cytotoxic T cell population. So let's summarize again the same thing which we have discussed. So these are actually master transcription factor which are regulating the CD4 plus differentiation, and that is what the three factors which are listed here. I just wanted to emphasize one more point here, that GETA3 and Icaros protein which we have discussed in the central label generation of this T lymphocyte is actually a zinc finger transcription factor. So GETA3 and Icaros is actually a zinc finger transcription factor. What does it mean? That zinc finger transcription factor are, suppose these are the proteins which are stabilized by the addition of zinc ion, and when zinc is involved in stabilizing this molecule or a protein, then these are actually called as zinc finger transcription factor. Why it is called finger? Because their structure, the protein structure looks like finger, and therefore when people solve their structure, the protein structure, they look like finger, and they require zinc to stabilize their structure. Therefore they are called zinc finger transcription factors. So the Th2 differentiation is taking place by a zinc finger transcription factor which is GETA3. And these are some of the things which we have already discussed. So let's try to look into how things are happening in the cytotoxic T cell or CD8 plus T cell population. So the cytotoxic T cell transcription control is happen in a different way, but the initial cytokines are very similar like Th1. So in Th1 also we could see that IL-12 and type 1 interferon, particularly interferon alpha and beta is involved here, while in case of the Th1 it was interferon gamma. But type 1, generally when we discuss about interferons, so interferons are actually multiple type, but basically are largely explode into a interferon type 1 and type 2. Type 1 is alpha and beta, and type 2 is gamma. So alpha and beta is actually type 1 interferon and gamma is type 2 interferon. So when we are talking about the development of or the transcription control of the cytotoxic T cell population, then IL-12 and type 1 interferons are particularly playing a very important role, which are interferon alpha and beta. Now this can cause the expression of two transcription factors, same like Th1, T-bit is coming here, and EOMES, which is another transcription factor very critically controlling the expression here, is expressed in case of differentiating CD8 plus cell. And after getting this transcription factor binding to the DNA, we get an expression of granzyme, perforin, interferon gamma and KLRG1. So these are very critical protein which are produced upon the activation of CD8 plus, and this further differentiate this particular cell to a cytotoxic T cell population, which is very important in killing the infected, killing very specifically to the tumor cell. So we looked into this EO-MES transcription factor, which is actually eomesodermine, is a critical transcription factor belonging to T-Bax family, same like a T-bet, and it plays a fundamental role in vertebral embryonic development, particularly to the immune system aspect. And then apart from that, we have these proteins which are getting expressed once this transcription factor is binding to the cell. We see that we have killer cell lectin-like receptor G1, which is called KLR-G1, and these are surface receptor, often considered a marker for late stage differentiation, particularly senescence or chronic stimulation of T cell and natural killer cell. So basically, when we call it a killer cell lectin-like receptor, definitely these are expressed in the cell which are known to kill a specific cell type. So these are expressed either in natural killer cell or in chronically stimulated T cell in respect to the CD8 plus T cell population. Again, we have looked into the killing effect of the CD8 plus cells, and CD8 plus kill the cells which are infected with pathogen by producing a specific kind of pore in the structure, and for that you require a protein called perforin. So these perforins are expressed in this particular cell type, and also granzyme, which can activate multiple proteins which can lead to the cell death, particularly apoptosis. So granzyme and perforins are also expressed by CD8 plus cell. Apart from that, we also see sarglycine, which are expressed particularly in this cell type. That means when we are talking about the cells which are going to do a function, we require to produce all those proteins which are required to do functionality of that cell. And since CD8 plus T cell will directly kill the cell, we require to have a production of perforin, granzyme B, and also sarglycine. So these are three. Apart from that, we have killer cell lectin-like receptor also expressed in this particular cell type. So this is about the cytotoxic T cell population transcription control. So we looked into T helper, Th1, Th2, Th17. We also looked into CD8 plus cell. There is another cell type which I started discussing, is called T regulatory cell population. And the T regulatory cell population is actually a very important cell which control the immunosuppressive activity, particularly towards the tolerance when we talked about reactivity towards the self antigen, and also to control the excessive production of the immune response. So these cells are regulating the expression of the T cell function. We will see how these cell type can do function, but let's focus here that these are very specific cell types. So these are the cells which are actually a T cell, but they express a very specific protein called FOXP3, and they can secrete different cytokine, particularly IL-10 and TGF-beta. So these TGF-beta and IL-10 are very specific cytokines which are secreted by this T regulatory cell. Now, when we say that these two cytokines are secreted by T regulatory cell, a very obvious question is what could be the function of this, and the answer is this could be an immunosuppressive. So these are immunosuppressive kind of cytokines that is produced by T regulatory cell. So TGF beta and IL-10 is immunosuppressive in nature. They can suppress the specific immune response, and the T regulatory cells are positive in FOXP3 expression. Now let's try to understand what is the mechanism, how the T regulatory cells are actually controlling the immune response. So let's see, there are multiple, we'll go one by one. So we'll try to focus on the A one. So how this FOXP3 can control the dendritic cell population. So we have understood that CTLA4, which is cytotoxic T lymphocyte antigen 4, which is again immunosuppressive in nature, which can be a negative signal towards the proliferation, and also LAG3, which is called the lymphocyte activation gene 3, which is also called as CD223. These are getting activated and expressed by FOXP3. So one has to remember that what are the things which are expressed on the surface of a T regulatory cell. Of course, these are FOXP3. That means there is an expression of FOXP3 in the surface. So this FOXP3 will be there, and apart from that, we are getting an expression of lymphocyte activation gene 3 and CTLA4. So these can bind to the CTLA4 can bind to CD80 and 86 co-stimulatory molecule, which we have discussed earlier, and LAG3 can bind to the MHC2 specific molecule. And once there is a binding, we could look into the metabolic changes which can happen in the cell. And one such change is towards the tryptophan metabolism. So in the tryptophan metabolism, the tryptophan can be converted into multiple steps into kynurenine, and this can be done by IDO1. So IDO1 is a very specific IDO1 is a very specific enzyme which can help and participate in tryptophan metabolic pathway to produce kynurenine. And kynurenine is actually the blocker of the T cell effector response. So essentially, this can block the T cell activation. So what we could understand here that the FOXP3 specific gene can downregulate the dendritic cell population by downregulating or modulating the tryptophan metabolic pathway by producing kynurenine, which can inhibit the T cell proliferation. Second is through extracellular vesicle. So the extracellular vesicle can also load multiple inhibitory factors which can cause an inhibition of the T cell effective immune response. And third is by immunosuppressive cytokine, and which we looked into in the last slide, that TGF beta and IL-10 is actually immunosuppressive in nature. So once these are going into the circulation, they can downregulate the T cell immune response. Next is they can also do very specifically cellular cytolysis, and the cellular cytolysis can be done by the expression of the granzyme and perforin. So granzyme and perforin can be produced by these FOXP3 positive cells, and they can go through this pore and can dissociate the cell by apoptosis. So basically cell death is happening because through these pores the granzymes can enter, which can activate the apoptotic pathway by activating multiple genes, because we have looked into these are caspases, so they can cleave the protein to activate downstream protein which are involved in apoptosis. The other very important pathway is metabolic perturbation. So essentially FOXP3 can do multiple different ways to suppress the immune response, and one such other method is by doing problem in metabolism. So they can express a receptor called CD25, and this CD25 is actually scavenging the IL-2. Once the IL-2 is scavenged by the CD25, the IL-2 is not available for T cell proliferation, and there could be a death of the T cell. So what they are doing is basically they are challenging, they can engulf all the IL-2 through their receptor called CD25, and therefore there is no IL-2 available to do proliferation of the cell. We have understood that IL-2 is actually a very specific cytokine which are involved in the proliferation of the T cell and B cell. So these are basically killing the cells because there is no cytokine. So we can say that there could be a cytokine starvation which can lead to the cell death. And the other reprogramming which they are doing in terms of metabolism is basically they can do a passive transfer of the cyclic AMP to the T effector cell. So T regulatory cell can do cyclic AMP transfer through the gap junction to the T effector cell, and this can lead to the very specific expression of adenosine receptor which can bind to the adenosine, which are produced by these two receptors CD39 and CD73. So the FOXP3 cell can express CD39 and 73, which can convert adenosine triphosphate to adenosine monophosphate and adenosine monophosphate to the free adenosine molecule, and this adenosine can go and bind to this particular cell, which can lead to the T cell immunosuppression. So these are some of the documented, well proven pathway which can lead to the T cell suppression. by FOXP3 positive cell, which are called T regulatory cell. So these are the mechanism of T cell suppression by T regulatory cell. So let's try to recapitulate one more time. What are the things which are involved here? So the first thing is suppression of dendritic cell by modulating the tryptophan metabolic pathway through the extracellular vesicle. They can do an immunosuppressive cytokine production such as IL-10 and TGF-beta. They can also do a cytolysis by specifically producing granzyme and perforin, and also they can do a metabolic perturbation by scavenging IL-2, causing IL-2 starvation to the cell type, leading to their cell death. And also they can convert the ATP to adenosine by the expression of CD39 and CD73. So because of this, the adenosine can bind to adenosine receptor, which are expressed by the expression of cyclic AMP that are transmitted to T effector cell by T regulatory cell population. So essentially these are the ways T regulatory cell can control the immune response, and that we will try to understand why it is important, because one way we are trying to understand there could be an immune response, and the other way we want to understand that how we can suppress the immune response. So this is how the mechanism of T regulatory cell happens. So now we have understood everything in terms of T cell transcription control. So Th1, Th2, Th17, cytotoxic T cell and T regulatory cell population. Now what will be the control of the activated T cells? So once the T cells are activated, all those 5 different T cell type, we got an activation. Again, there could be a controlled transcription of the activated T cell population. So let's try to understand what could be the transcription control of the activated T cell. So these are controlled by 2 transcription factors. One is called interferon responsive factor or regulatory factor 4, and MYC protein. So these can be induced by TCR signaling and some other controlled way, but just for knowledge of how these are controlled, that we have 2 transcription factors which are controlling the activated T cell population by doing metabolic reprogramming. What do you mean by metabolic reprogramming is oxidative phosphorylation of the naive T cell can move to activated glycolytic process. So oxidative phosphorylation is an oxygen dependent process, while glycolysis is kind of an oxygen independent process. We called it anaerobic process, and in the next slide we will try to understand what are the difference between the oxidative phosphorylation and glycolysis in terms of metabolic reprogramming. So we can see that the metabolic reprogramming can happen, and the ultimate outcome is cytokine production, proliferation, and the cytotoxicity towards the pathogen. So let's try to see what are the c-MYC, specifically L-MYC and N-MYC. So these are the family of regulator gene which are actually proto-oncogene. They can help in the proliferation of cell type. So c-MYC is actually most explored one. It is activated in various type of cancer cell. L-MYC is particularly involved in small cell lung cancer, and N-MYC is particularly for the neurons. So these are heavily expressed in neuroblastoma and particularly neuroendocrine tumors. So these are basically the classification of MYC which we are talking about. So these are the control of activated T cell population, so which are controlled through IRF4 expression and the MYC expression. This could be c-MYC, I-MYC, L-MYC and N-MYC expression. Now since we started with the metabolic reprogramming, let's understand what do you mean by metabolic reprogramming, and particularly in context of T cell. So these are actually moving of one type to the other type. So say one type of phenomena can be changed into other type. So we'll see what are the difference between glycolysis and oxidative phosphorylation, because what we learned that oxidative phosphorylation is moving towards the glycolysis in T cell activated T cell signaling cascade. So glycolysis is actually a process that happens in the cytoplasm, cytosol, while oxidative phosphorylation takes place in inner mitochondrial membrane. The glycolysis, as I said, it's an anaerobic process, doesn't require oxygen, whereas oxidative phosphorylation is an aerobic process which requires oxygen. Glycolysis mostly produces a very small amount of ATP, 2 ATP, while oxidative phosphorylation in combination with the Krebs cycle can produce approximately 28 to 32 ATP per glucose molecule utilized. So this is more energy producing process. Glycolysis is very, very fast as compared to oxidative phosphorylation. So it's a very fast process. The end product of glycolysis is 2-pyruvate, 2 ATP and 2 NADH. So these are actually the end product of glycolysis. While in oxidative phosphorylation, we see a large amount of ATP produced at the end of this oxidative phosphorylation. Glycolysis provides a fast energy and intermediate for biosynthetic pathway, while oxidative phosphorylation is primarily for efficient energy source for resting and aerobic cells. So basically, this process is activated when we have more of the oxygen in aerobic cell, while glycolysis is a cytoplasmic, very specific process that happens in absence of oxygen. So these are basically reprogramming effect which we have understood that's happened in the activated T cell in order to help them to proliferate and do a function. So finally, we looked into different transcription factor and regulation of T cell. Let's summarize that in one slide. So we could see that the Notch1 E protein and Ikaros1, these are controlling the transcription level at the thymic central level. Then we have T-bit, GATA3 and ROR gamma, particularly these are controlling the peripheral signaling process, the transcription control in the peripheral level, Th1, Th2 and Th17, while the activated T cells are controlled by very effectively IRF4 and MYC expression in the cell. So this can actually do all the function which we have discussed. So these are stage specific role which I am summarizing here, the same thing which we have discussed till now. And finally, what we have understood that there is a coordinated effect in the T cell expression. So we have hematopoietic cell, Th1, Th2, Th17 and finally the cytotoxic T cell population. So there could be a series of events that can happen in the cellular proliferation, and these are mostly governed by the expression of the transcription factors. And we looked into those transcription factors very critically in both B cell as well as T cell. So what we can get a take-home message from this module is basically how this transcription factor critically regulating the expression of the gene when they are transmitting themselves to the nucleus. Sometimes it is dimeric form such as STAT protein, sometimes it is a monomeric form such as NF-kappa B, and we see that they can modulate different cytokine in a variety of different ways. So this is all about the transcription control of T and B cell, and we'll try to look into some other aspect of the immune cell in our next module. Thank you.